Hi everyone. My name is Alex. I'm going to start off with a quick background before we get into the interesting stuff. So I was born in Phoenix, Arizona. I was an extremely energetic child. You could constantly find me outside hunting for critters. So I was absolutely thrilled when my family decided to move to rural Idaho, where I was constantly surrounded by tons of animals and plants. Here's a photo of me at the rodeo with an absolutely fantastic hairdo. And then many years later, I got my first introduction into textbook biology when I took AP Bio. And I found it to be an extremely difficult course, but I was really inspired by understanding life at the molecular level. So I didn't let my struggles get to me, and I decided to pursue a Bachelor's of Science in Biochemistry at Western Washington University, which is north of Seattle, just below the Canadian border. It's an absolutely beautiful place if anyone is considering going to the West Coast for college. Here's a photo of my fantastic lab group, and I'm over here in some really bright colors. Um, and then I just loved doing research, working with my hands, and running around the lab. So I decided to pursue more research and school for the next 6 years of my life. And I moved all the way to the East Coast, where I'm currently in my third year of a PhD program in biochemistry and biophysics at Brandeis, which is right down the road. Um, so yeah, from Idaho, I moved directly to Washington, and then hey, the dog barked for a second. But in college, I joined a biochemistry research group, um, and it was an incredibly exciting experience. I made wonderful friends and felt very passionate about the research we were doing. So we were really interested in understanding how blood forms a clot. But what is blood clotting? So after you get a cut, normally your body will adjust and form a scab. But sometimes mistakes in our DNA can lead to issues in the proteins. So DNA is a molecule that carries all of the code for building and maintaining an organism. Occasionally, alterations in our DNA will lead to problems in the proteins. So this is shown in patients with hemophilia, which is a rare inherited bleeding disorder that prevents the body from properly folding or forming clots. And it's caused by a deficiency in this clotting factor, factor 9. And factor 9 is a protein. So without a working factor 9, people with hemophilia are unable to properly form clots. So the treatment for this is that patients will receive blood injections that supplement this missing factor 9 several times a week. And it's a life-saving treatment, but it's absolutely inconvenient. So what if there was a better treatment? What if we could make a factor 9 protein that was super active and lasted longer, requiring less treatment? So, as it turns out, while I was working in the lab and we were studying blood coagulation and this protein factor 9, a paper came out from a different lab that identified this protein with enhanced activity, meaning it was like a super protein. And the way that they found this protein was using a technique called ancestral sequence reconstruction. So when I saw this paper, all I understood is that ancestral sequence reconstruction is getting the DNA of ancestral sequences and reconstructing it. And my understanding was that it results in proteins that have properties that are different from modern day proteins. But really, how does this work? How do scientists do this process? And what's needed? I was extremely fascinated by this idea of ancestral sequence reconstruction and studying proteins from ancestral species that are no longer alive today. So um, I interviewed at schools across the country, and after I decided I wanted to go to grad school. But my current boss studies ancestral sequence reconstruction. And so I moved all the way from the tippity top of the West Coast to the East Coast to really just answer the question, what is ancestral sequence reconstruction? So now I'm in a lab that tries to understand how these methods work and the best way to resurrect and study these ancient proteins. So, in ancestral sequence reconstruction, the goal is to figure out what the DNA or protein, like factor IX, looked like in ancestors that are no longer alive. And as an intro, I kind of wanted to touch on the idea of a family tree. So, a few years back, I sent my dog Hank, who was barking a couple minutes ago, I sent his DNA into this website Embark to try to identify what his ancestors were. So I got Hank at a tractor shop in Idaho. So I was pretty curious what he was. I mean, he kind of looks like a border collie, but he's way bigger and fluffier than most border collies I've seen. So, by looking at the DNA of a bunch of different species, you can figure out what the ancestors probably looked like. And this is sort of Hank's lineage that they predicted. He has some Bernese Mountain Dog, Australian Shepherd, and then less Border Collie than you'd actually predict, or that I was thinking. So, ancestral sequence reconstruction is a lot like building a family tree, but instead of whole organisms, we're looking at the DNA that codes for these specific proteins like factor IX that I've been talking about. So we just want to figure out the oldest version of what these proteins looked like. And after working with factor IX protein and thinking about applications like this, I just have become fascinated with the idea of ancestral sequence reconstruction. I mean, we have the power to look at ancestral proteins and understand how things evolve over time. So, why does it matter? All organisms evolve over time. Even your DNA is probably different than your great-great-grandparents' DNA, just because small mutations happen over time that will gradually change how a protein acts or the things like that. But this process of ancestral sequence reconstruction or looking at lineages comes with problems. So, just like if I sent Hank's DNA in today, I would probably get a slightly different answer, or maybe a refined answer than the one I got 4 years ago when I sent his DNA in. This is because our methods and techniques for predicting and understanding how DNA changes over time are constantly improving. So the research I do now is focused on how do we predict the best ancestor. but why would we need to predict the best ancestor? So maybe some of you have seen the Jurassic Park movies. And I thought this might be a good analogy if you've seen it, but I'm not sure. So in the movie, they are trying to resurrect dinosaurs, which obviously are not around today. And they're using just snippets of DNA that they found. So in ancestral sequence reconstruction, we're doing the same thing, but on a microscopic scale. So rather than resurrecting the whole organism, we are just resurrecting a little piece, which is a protein like factor IX. So one thing I really just want to point out here is I love this image because it compares the Jurassic Park Compsognathus versus what current science tells us the real Compsognathus dinosaur looked like. And one thing you'll notice is that the real Compsognathus, based on current DNA technology, suggests that it had feathers, which was very likely an important part of the dinosaur and super important for understanding how other things have descended from these dinosaurs. So yeah. My current research is just trying to understand what was the true ancestor like. And are our ancestral protein resurrections or reconstructions accurate? Or are they possibly artifacts of this methodology, which is a prediction that we're using? So it's possible that the factor IX protein that was identified as super was actually more active than the modern day factor IX protein. It's possible that a long time ago we needed this protein for a higher ability to clot blood. But it's also possible that it's just an artifact of the methods that we're using. So just like in Jurassic Park, we might not have the methods for resurrecting ancient DNA and protein very accurate. But if we want to understand as scientists how things evolve over time, it's best that we resurrect the most accurate ancestor. So that's what I'm working on, and I hope you enjoyed this, and I will happily take any questions. I'll just end this with my dog Hank, the Australian Shepherd Border Collie Bernese Mountain Dog in a very messy lab. And then my current lab mates, some of my friends that I work with at Brandeis, and some protein crystals. Thank you so much.